extracellular matrix goes wrong. But one of the things we're looking at that's especially important in development is the signaling roles of extracellular matrix. Because cells are embedded in this stuff, or cells that migrate from one part of the embryo to another part of the embryo will end up being in contact with extracellular matrix produced by other cells. And that actually has a signaling function. It can affect cell differentiation. It can affect development. In fact, contact with extracellular matrix is a major form of cell-to-cell communication. Now, just a little history behind this. We mentioned, and this is how they discovered the existence of growth factors. We mentioned that when people tried to culture mammalian or human cells, they couldn't do it, and then somebody came up with the idea of adding some blood serum from animals, and then some cells could indeed be grown in culture. But the only cells that they could successfully grow in culture happened to be derived from extremely aggressive cancers. Things that spread and proliferate like crazy. But normal cells, if you just get out like a tissue sample or something, they still couldn't get going. If you took cells, put them in a petri dish, threw in the appropriate serum or growth factors or whatever, the vast majority of non-cancerous cells would simply round up, float around, sit there staring at you. Metabolism would be low. They wouldn't divide, and eventually they would die. So there was something missing, that most ordinary cells required something over and above those growth factors that they added in the serum. The only ones they could do in just serum and culture media were derived from very aggressive metastatic cancers.
Is it something to attach to?
Ah, you got the right idea. Well, here you have your petri dish, glass or plastic, and your culture media, and the cells are just floating around. Well, people thought, okay, hey, in real life, these cells are constantly in contact with extracellular matrix that they secrete and nearby cells secrete. So what if the case, what would happen if there's something in that extracellular matrix that is essential for normal cell behavior? So what they decided to do was, before you put the culture media in, they threw in a thin layer of fake extracellular matrix. It was usually a mixture of collagen and fibronectins, and sometimes they threw in some polysaccharides, made up little solutions, poured it in the bottom of the culture dish, let it kind of dry out, then put your culture medium in, and lo and behold, what they found was the cells would settle on and attach to the surface. And then they would sprawl out like a fried egg. They would actively start moving around. And most important, their metabolism would go up to normal levels. And most importantly, they would actively divide. Yes?
ECM what?
Signaling.
Signaling? Okay.
Okay. So that's telling you, as the contact with the extracellular matrix must be doing something to the cytoskeleton, because it's causing cell shape changes. It's causing cells to attach to the surface. They wouldn't attach to the glass or plastic, but they would attach to this fake ECM. So we're affecting the cytoskeleton, stimulating cell movement. It's affecting cell metabolism, ramping it up. So in other words, we must be affecting numerous enzymes. It's causing cell proliferation, cell division, so it must be doing something to stimulate cell division. And depending exactly what is in that extracellular matrix, in some cases you could put immature embryonic cells in, and just by altering the ECM composition, those cells could differentiate in one direction or in another direction. So this could stimulate cell differentiation, which means changes in gene expression as well. So, this stuff must be doing is doing a lot of things to cells that are in contact with it. Now, you can clearly see that the extracellular matrix materials, the proteins and polysaccharides, are not actually taken into the cell. They're on the outside of the cell, and they stay on the outside. So whatever is going on, the cell is not taking these things into the interior of the cell. So what that must mean is that the extracellular matrix is activating one or more signaling pathways to give the results that we see. So that's an important thing. The ECM is a signaling agent. And instead of being soluble, secreted molecules, it acts like a solid-state signaling agent. But it does important signaling functions nevertheless. Okay, now, the question is, how does this signaling work? All right, well, we have a pretty good idea about the pathway now. And you notice one thing it does especially, stimulates cell division and cell differentiation. Do we know of some kind of signaling pathway that does that quite a bit? Yeah, certain growth factors. A lot of growth factors do that. Many growth factors use the RAS-MAPK pathway. And in fact, much of the signaling is indeed due to that. So what we're going to do here, first of all, we need some Ecm receptors. We need receptors that will bind to the extracellular matrix. And we have a big family of proteins. Oh, especially, spell it right. And these are called the Integrands. The Integrands are large proteins made of an alpha and a beta subunit. There are many different kinds. They're very large proteins. They go through the membrane, but they stretch long distances both in and out of the membrane. So here we have our Integrands. Each integrin has multiple binding sites on the outside for different ECM components, each type of integrin. And depending on the integrins you're doing, that determines what kind of ECM components they bind preferentially to. For instance, some integrins have a lot of binding sites for different types of collagens, but maybe not for some of the polysaccharides, or maybe not for fibronectins or laminins or what have you. Others may prefer certain fibronectins and not bind certain collagens much. Some will bind certain types of collagens very well and bind other types very poorly. Some will bind certain polysaccharides especially well, but not other things. So there are many different types of integrins, and each one has its own preference. It has its own preferential binding sites, things that it will bind to. So we can have a whole bunch of different integrins, each with a different preference for some kind of ECM components. Now, when the integrins bind, yes?
What's the bottom word you just wrote?
Components. Whether it be proteins, polysaccharides, or whatever. Now, when the integrins bind, whatever the appropriate ECM components are, they're going to change their tertiary structure inside and activate several different things. We're going to have to look at these guys one by one. First, let's look at the signaling thing. What they do, and apparently this is an indirect thing, the first thing that happens is they activate two protein kinases, one called FAK and the other called SRC. FAK is called, it's often referred to as focal adhesion kinase. And SRC stands for sarcoma. Those are soft tissue tumors that you find in different organisms. They're both protein kinases. Now, these guys phosphorylate a number of things, but some of their main targets include the phosphorylation and activation of the guanine nucleotide exchange factors that in turn stimulate the GDP and GTP exchange in our old friend RAS. And when we do that, that means we have activated RAS in the GTP state. And then, of course, these activate the RAF kinases and the MAPKKs. And our third set, the MAPKs, the third set of kinases. Lo and behold, cell differentiation, cell division. In effect, the extracellular matrix acts like a growth factor because it hijacks, using a somewhat different way, it activates the same RAS MAPK growth factor, signaling pathway that many growth factors do. So in some respects, the ECM can act like a growth factor, stimulating cell division and differentiation in the cells that it comes in contact with. Now, whether that happens or not depends on whether that cell is in contact with the kind of ECM composition that stimulates the integrins the most. If you have integrins that don't bind whatever ECM you're floating around in or in contact with, nothing will happen. On the other hand, if you do have integrins that bind particular ECM composition, you're going to get some kind of effects. So that's one kind of thing. We hijack and activate the RAS MAPK pathway. So that's an important role of how these things work. Now, just a little historical thing here. Kind of neat history. We look at this guy, Sark. Okay, how did we find out about it? Well, many years ago, you go back to about the late 1960s. Richard Nixon got in power, and there was a big thing, and this was some politics, but the idea of one of Nixon's projects was starting what he called a war on cancer. He said, you know, we can fly people to the moon and cancel most of the Apollo program shortly afterwards, but if U.S. can go and do a project like flying people to the moon and getting them back in one piece, why can't we put U.S. ingenuity and science to go after cancer? Okay, so the idea, created a new federal agency, a subset of National Institutes of Health called the NCI, National Cancer Institute. And a whole bunch of money, a couple $1 billion a year was going to go into it. The problem is he didn't include any extra funding for that, so that came out of other NIH money, so I was robbing Peter to pay Paul. But when they started the National Cancer Institute, at that point, especially some of the physicians and scientists that were heading and very influential in that program, there was the hypothesis that the majority of cancers were caused by viral infections. And that's what they're using as the operating assumption. And they put lots of money into looking for cancer viruses, and not as much money into some of the other aspects of it. Okay, so here we are back 1970s, early 1970s, and they did indeed find animal and human cancer viruses. Along come the ability to do gene splicing, recombinant DNA, where you can take and cut out sections of DNA from one organism and put them into bacteria, get the genes expressed, get enough copies of genes so you can sequence them and things like that. All those tools of molecular biology that are so widely used nowadays. Okay, well, one virus they found is something called the RSV, or Roux sarcoma virus. Roux was in the late 1800s, a classic embryologist. He did a lot of basic embryology, doing experiments carefully, painstaking experiments under a microscope and stuff like that. But he had also discovered that these kind of soft tissue tumors called sarcomas, in birds like chickens and stuff like that. And he noticed that these things would form, and they were clearly abnormal and highly proliferating cells, and eventually it killed the bird and stuff. Well, years later, they identified this as a virus. Those cells were infected with this particular virus, so they called it the RSV. Okay, well, when they looked at the virus, they found this virus had a mere 4 genes. And with the tools of molecular biology, they would take this virus and they would successively delete genes one by one and ask what genes are essential for viral replication and viral spreading, and what genes are actually cause the cancer. Well, they found 3 of the 4 genes were essential for viral replication. If you deleted that, the virus could not infect cells and it could not replicate. But one of the genes, if you delete the virus, could still infect cells and replicate quite normally and spread and stuff. But what it could not do anymore is cause cancer. So these folks found a cancer-causing gene, or oncogene, the first gene, oncogene discovered. Now this is down, we're getting to about 1974, 1975 here. So it's like, wow, we have a cancer gene. This is going to be really important. So at a conference, a big nationwide conference, some of the people that did a lot of this work had proposed, we want to find out what this thing does. So what we're going to do is we're going to take the gene from this and we're going to put it in our little microbial, widely used pet organism for genetic engineering, E. coli, the same bacteria that's found in your digestive tract. And they're mentioning they're going to splice this gene, which they call SARC for sarcoma, into E. coli in the lab. It's make enough and see what this thing does. And then a lot of people said, whoa, hold it a second. Great idea, good experiment, but bad idea. You are going to take a cancer gene. You are going to, a known cancer gene. You are going to splice it in the very same type of bacteria that's found in your own digestive tract. Does this sound like a good idea? No. So let's call it, then a whole bunch of scientists called a moratorium on genetic engineering experiments, gene splicing, until there was a better assessment of the potential risks and hazards and stuff. And then the federal government got in and set a whole bunch of rules. A number of countries did the same thing. And someplace it got to be a political hot issue. Harvard University had a real fight with the community on their hands because they were going to set up a lab that was going to do a lot of what is nowadays routine molecular biology. And there was, there were fears that they're going to create the Andromeda strain stuff. And there are all kinds of federal regulations. There still are, but they eased up a lot. About the only kind of recombinant DNA experiments are where you're splicing genes from known nasty pathogens, stuff like that. And there's strains of E. coli that really cannot survive out of the lab, and those are widely used and stuff like that. And finally was the clincher. They decide to deliberately test out the potential risks. So they splice some nasty genes into regular E. coli in an old germ warfare lab in Fort Detrick, Maryland. And one thing they found is not only is it fairly difficult to get eukaryotic genes expressed in bacteria, but the other thing they found is, I guess you could say, evolution in action. Those bacteria, if they were released into a simulated environment, they would quickly get outcompeted by the unengineered or wild-type bacteria. Even if they could survive outside the lab, the normal bacteria would outcompete them. And then that's the reason why. If you're putting in and getting a bacteria to express a gene that does not contribute to its survival, it's going to be a slight disadvantage, maybe minuscule, 1 part out of 1,000 or 10,000, to the normal strains of bacteria. At minimum, you're wasting energy to make these molecules, and you're wasting amino acids to make these proteins and stuff like that. Puts you at a slight disadvantage. And when you're dividing in a race where you're dividing every 20 to 30 minutes, it doesn't take too many generations for the unengineered bacteria to win. So that turns out that evolution kind of saves us. So once that data was in, they downgraded a lot of them. The vast majority of recombinant DNA experiments, you're just doing a standard microbiology hood. I mean, several people in our department do that. A number of students do that kind of stuff. And only certain experiments have to be put in higher levels of containment and stuff. So, okay, that's what we had there. But eventually, they finally did that experiment. They got that gene, the SARC gene, expressed in a model organism. Yeah, it was a genetically crippled E. coli and under high containment, all this kind of stuff. And they found, what is that? And they sequenced the gene, and they got enough of the protein expressed to see what it did. It was a protein kinase. It phosphorylated other proteins, but it was a weird protein kinase because it phosphorylated proteins on tyrosines. It was a tyrosine protein kinase, the first discovery. Ordinary kinases stick phosphates on serines and threonines, but this one put it on tyrosines. That generated a huge amount of interest in looking for other tyrosine kinases because the first tyrosine kinase discovered causes cancer, at least in these chicken and bird cells. And sure enough, they found other tyrosine kinases. And many of them seem to be potential oncogenes as well. So the question is, what were these tyrosine kinases doing? Second of all, if they saw that this particular gene happens to cause cancer in birds, do we have anything like that in our own genome? This is using the power of DNA hybridization, southern blotting. What they do is they make a probe to part of the viral SART gene and then see if it hybridizes to anything in our own genome, and lo and behold, it does. In fact, it does it to all, all vertebrates, and I think most invertebrates as well. Okay, so we have a SART gene. What's the difference? How come it doesn't cause cancer in us? Difference is the viral SART gene is missing a portion of it. It apparently, viruses often do this when their DNA gets copied. A lot of times it takes some host cell DNA along with it. Somewhere in the course of this virus's evolution, it took a portion of this SART gene, a major portion of it. The part that was missing was the one that allowed the kinase activity to be turned on and off. So in other words, the viral SART was active all the time. It was not regulated. It illustrates the importance of protein regulation. That's why I emphasize it all the time, right? Okay, so the thing was a continuously active kinase. Later on, they found some of these other tyrosine kinases were also different from the normal cellular versions, and that affected the regulation. So we found SART was the first tyrosine protein kinase. It took many years of research to find out what SART did. And one of the things it does is activate and fire off the RAS-MAPK pathway, which they did not know about in those days. But there was a lot of interest in tyrosine kinases, and a large number of them are indeed oncogenes when things go wrong, and most of them are connected to cell signaling pathways. So there was a lot of interest in tyrosine kinases as oncogenes, and sure enough, we find connection. And as a matter of fact, many cancers can be considered when good signaling pathways go wild, and in other words, they're improperly regulated, they're on all the time and stuff like that. So that was a kind of cool little historical aspect about that. And now we have an idea what it does. It's kind of tied in with the connection of the extracellular matrix signaling pathway. Yes, you had a question.
Yes. What's that R word? Kinase.
Oh, RAF. That's
Okay.
Yeah, you remember
That's
all
what I
RAF.
thought, but I just.
Yep. Okay. Well, that's only one of the things that integrins can do. Activate signaling pathways. But the other thing integrins can do is regulate the cytoskeleton, especially microfilament cytoskeleton.